the element itself. So if you go from 1 proton to 2 protons, you go from hydrogen to helium, and then so on. Right? So messing around with protons changes everything. Messing around with neutrons just changes the mass of that same element. And then messing around with the electrons, which is what we're particularly interested in, will go from a non-charged particle or element, an elemental thing like sodium I have up there, to a cation of sodium. So that's the chemical reactivity of sodium, or of an element. So I'm gonna try to restart this. So it's communicating, I just don't know if the projector's gonna work. Hope I didn't mess up the projector.
That'd be unfortunate.
That's what?
That'd be unfortunate.
Well, so here's the problem, is that we gotta take this quiz, right? So, if it's unfortunate, no, power. All right, so I just wanna warn you, I'm going, I have to go fast, because this is, but the, let's see.
There's 92 slots.
I guess that's even more reason to record it.
Were you already recording?
Huh?
You were-
Oh, I am.
All right. Good. Okay. So the chemical basis of life is the title of this chapter. If you look at the picture on the right-hand side, we have a couple of drawings here. The drawing that I have here is water molecules. These are water molecules. You can see these red dots right here are called, they're bonds, and I don't know if I mentioned what those bonds were when I drew them between adjacent water molecules, but there's little charges, positive charge here and a little negative charge there. That temporary bond between those adjacent molecules is called a hydrogen bond. This is one way to draw multiple water molecules. Here's another way to draw multiple water molecules. If you look at the top, we have examples of a nucleotide, and we have parts of a nucleotide. On the right-hand side of this, we have a pentose sugar with a phosphate group, which is right here. And then this is a nitrogenous base, and there's 2 rings. And this over here is a nitrogenous base, and there's 1 ring. So these 2 rings are wide, this 1 ring is narrow, and the distance between this is constant. There's also hydrogen bonds right here between them. So hydrogen bonds is a chemical bond that's very, very important in this. When I talk about I'm not sure why this looks like this. The periodic table, we're going to be referencing this periodic table. And here's what it looks like. Again, I talked about the first 2 columns, columns 1 and 2. Those form cations of either +1 or +2. These columns 15, 16, and 17 will form, sometimes they form anions. Generally, they also form covalent bonds, which is what we've been drawing. But the charges on fluorine and chlorine will be -1, as I have up there, with chlorine. Oxygen will have a -2 charge, and so on. So if you're over on the right side, you're trying to gain electrons. If you're over on the left side, you're trying to lose electrons. Again, all you're trying to do is to achieve the stability of the right-hand column right here, which is the noble gases and the inert elements. So basic chemistry, let's just go through this very, very quickly. First of all, if I ask any questions about basic chemistry, atomic mass, the name of this element, the number of protons, the number of neutrons, etc., you'll have this in front of you, okay? So, if you talk about mass, atomic mass is just simply the sum of the protons and neutrons in the nucleus. Electrons don't have a mass. They're very much weird things. The entire electron structure occupies space. So electrons don't have mass, but they occupy space, which is weird. When you're talking about elements and atoms here, they're the simplest molecule or simplest things that we have in our bodies. Have we talked about hydrogen, carbon, nitrogen, and oxygen? Yes, we have. Have we talked about the 4 macromolecules? Yes, we have, and that's what we're going to preview already. So what I've done with the discussion yesterday and earlier today is basically give you a heads up as to what I'm going to talk about in this lecture. If I don't finish it today, then it's your job to go through it, right? Or if I skip over stuff, it's your job to go back to that. If you look at some of these elements right here that I've listed with the elemental formula in parentheses, these are some examples of some of the elements that we'll worry about over the course of the semester. The 2 that is super important is the sodium-potassium bond. If you notice this K plus and this K, or this Na plus and this Na, is what we talked about earlier today with the chemical reactivity. Elemental sodium, elemental potassium, those do not occur in our body. Ionic forms do. So those ionic forms are dependent upon the 3 subatomic particles, and really just one of those 3 subatomic particles, the electrons. Electrons have a -1 charge, and so if you lose 1 electron, don't get confused. If you lose an electron, then you gain a charge, you increase your charge. If you gain an electron, you decrease your charge. Neutrons and protons do not move, and that's when you talk about atomic bombs. Atomic mass and atomic number. Mass number, right, that's pretty straightforward. That's something in basic chemistry. When you start messing around with the number of neutrons in the same element, I said earlier, if you mess around with the protons, you change the element itself. You mess around with the number of neutrons, you have something called an isotope. In this case here, we have the most common naturally occurring hydrogen on the left. In the middle, we have deuterium, and on the right, we have tritium. Those that name D, or the prefix DEU means 2, TRI means 3. So the atomic mass of this element right here is 2. The atomic mass of this element is 3. The atomic mass of this one on the left-hand side is 1. This is the most common form of hydrogen, and so these are all naturally occurring hydrogen isotopes. Other elements have other isotopes, and regardless of the isotope, you're just messing around with the number of neutrons. I don't think that I talked about Avogadro's number here in the class. You might see a question on this, but that's basically, Avogadro's number is just basically how many atoms are in a given what's called a mole of that element. There's a whole bunch of atoms. How many atoms? 6.0222 times 10 to the 23rd. That's a very, very big number. Two basic bonds. There's really 4, but the 2 basic bonds are ionic and covalent. The difference between ionic and covalent, we've already seen. When you transfer an electron from one to another, and therefore you make an ion, here, that's an ionic bond. One example of that is sodium and chloride, or chlorine. The formula, there's sodium chloride. So that is an ionic bond, the transfer of electrons from one to another. Sharing of electrons occurs in covalent bonds, and you can have different types of sharing, we'll talk about that, and I've already mentioned it. When you start trying to fill up your outer shell of electrons, either by adding new electrons to your outer shell or losing electrons from your outer shell, like I just drew over here in sodium, that's called obeying what's called the octet rule. The octet rule means that, generally speaking, you want to have 8 electrons in the outer shell. And if those 8 electrons are in the outer shell, then that atom then, or that element then appears to be inert or non-reactive. When you talk about moving of electrons here, when you're talking about moving electrons, it's based on how chemically electronegative these elements are. So if you have a carbon and an oxygen, or you have carbon and a bunch of hydrogens. In the first case, you have carbon and an oxygen, you have unequal sharing of electrons, because oxygen is highly electronegative. If you have carbon and hydrogen, that's equal sharing of electrons. So that would be on this side of the arrow. Relatively unequal sharing of electrons would be between carbon and oxygen, or oxygen and carbon, or oxygen and hydrogen. That would produce polar covalent bonds. And then when you get extreme examples of that, very, very different, then you have ionic bonds. So this is basically different levels or different extremes. Cations or cations, anions, all this picture right here is what I drew right there. And you should be able to recognize, now if you look at this picture right here where my pointer is, or this picture right there, and I were to white out the element in the center. In other words, I could put a little tag right there, I could erase this part right here, and say, what element are you looking at? You would use your periodic table and say, oh, this is sodium, what element is this? This is chlorine. Notice the name here, so this is chlorine, and this is chloride, and there's slight differences here. We're not gonna worry about those. This isn't a chemistry class, but there is a distinction. If you look at all of this list here, you have lists of cations and anions. Those cations are the ones typically that have the positives right here. What is the difference between the first one right here? This is calcium, an incredibly important element, our cation. You can see some of its significance, biological significance on the right, but it has lost 2 electrons. Sodium has lost 1. How do I know? I can draw the electron configuration. Sodium is right there. If I were to draw calcium, calcium would have 2 in the outer shell. Go down to the bottom right here. Sometimes these formulas are temporary molecules. So for instance, NH4, that plus is a cation, but I said to you earlier that nitrogen has 3 places for 3 chemical bonds. But once in a while, it grabs another hydrogen, and then if it grabs another hydrogen, that additional hydrogen produces that extra charge. That's a very chemically reactive molecule, so it's not very long for this world.
What do you mean once in a while? Like, is there a certain thing?
So if you're chemically reactive, then you're not very stable, and you want to make lots of chemical reactions. You want to exchange electrons, right? So this element right here, or this compound, ammonium, is going to try and grab an electron from somewhere. When it grabs that electron, it's going to transfer it to one of these hydrogens. That hydrogen is going to go away, and then you're going to get NH3. Okay, and so then that's a much more stable chemical. Similarly, if you look at this one right here, HCO3, Cl minus and OH minus. I drew OH minus today, and I said that was a hydroxide ion. Once again, if there's a negative there, that means that's an extra electron floating around there. If it's a single positive, like there, there's just 1 electron missing. 3 names for that: H plus, hydrogen, hydronium, and a proton. More of these. Most of these are very simple elements here. Here, this is the chemical formula for a phosphate group. And if you look at this is a 3 minus right here. I'm not going to ask you about that in my drawing. I just did a P with a circle around it. That's the phosphate group. So covalent bonds come in a number of different forms, and I talked about those already. I've drawn them in a couple of different ways. I've drawn one, single and double chemical bonds. Here's a couple of examples. In this case, we have equal sharing of electrons. Equal sharing of electrons occurs because there's the same electronegativity. If it's the same element, there's going to be equal electronegativity. So if you have hydrogen, two hydrogen atoms, they'll share, they each bring one electron to the equation. The combination of those two is hydrogen gas, or H2, and it's relatively happy. It's much more happy than it is if it had just one hydrogen floating around. So that is called a diatomic gas, or diatomic atoms. There are three examples of diatomic atoms here. There's 1, 2, 3. Hydrogen. How many chemical bonds does hydrogen make? 1. How many chemical bonds does oxygen make? 2. And how many chemical bonds does nitrogen make? 3. So, hydrogen gas, or diatomic hydrogen, makes one covalent bond between two adjacent hydrogen atoms. Diatomic oxygen makes a single or a double bond, pair bonds, a double bond between two adjacent oxygen. And then nitrogen gas makes a triple bond between adjacent nitrogen. Okay, so there's 1, 2, 3. And this is all, well, to a certain extent. There is a little bit of hydrogen in the atmosphere. There's about 20% oxygen in our atmosphere, and there's about 78% oxygen of nitrogen in our atmosphere. Nitrogen, when we breathe it in, and then we exhale it, and we don't do anything with it. It's a very, very stable molecule. Can you recognize or draw this second compound, CO2 or carbon dioxide? Now, the way that I have it written, CO2, is not the way that it's actually drawn. First of all, it's in a line, and the line would be OCO. Each C has a double bond on either side of it, and that double bond goes to one oxygen, the other double bond goes to the other oxygen. Again, 4 chemical bonds for carbon, 2 chemical bonds for each oxygen. So can you draw the chemical configurations of this molecule? Can you identify this molecule? Once again, for tomorrow's quiz and for the exam, this will all on multiple choice. So you would see, I would have which of the following, what would the name for this be? Carbon dioxide. What would it look like? I could draw a number of different formulas here. And then one would be OCO with 2 double bonds. So in those cases, those 4 cases that had equal sharing of electrons. Equal sharing of electrons occurs between elements, because if they're the same element, you're not going to be stronger. One's not going to be stronger than the other. If you're different elements, but like in carbon dioxide, you can't, because of the structure of it, you can't share them unequally. Water is an example of unequal sharing, and I mentioned water yesterday, and today, water is incredibly important. When you see this little, well, first of all, this is the way that I generally will draw it, right here, where you have hydrogen on the left, hydrogen on the right, with a single bond between that, and oxygen in the middle. That oxygen is going to have a slight negative charge. That little D is a slight negative charge. That little D negative is a slight negative charge. That little D positive is a slight positive charge. What does that mean? It means that this oxygen steals the electron temporarily from this hydrogen, and this oxygen steals the electron from this hydrogen temporarily. So these delta negative or delta positive means that it's not a complete transfer of electrons, it's a temporary transfer of electrons. It's a covalent bond, it's a very stable molecule, and I'll give you an example of that. We saw a picture of taking sodium and putting it in water. If water wasn't stable, right? What would happen if I put it on a fire, right? So if there's a fire right there, and I pour water on it, it puts it out. There's an example of water being stable. If there's a fire and water is around and I put sodium or other elements in there, it would make it worse. There's an unstable molecule. So water is a very stable molecule, even though it's polar and covalent. I'm not going to talk about molecular mass. There might be questions about molecular mass. There might be questions about how you would get that question, but basically, where would you get these numbers? 22.99, 35.45. Where would you get them? The periodic table. If you look at this, this title here, intermolecular forces. We've been talking about, whether you realize it or not, intermolecular forces since yesterday. Intermolecular forces means it's forces between adjacent molecules. Some of them might be strong, some of them might be weak, some of them might be even, some of them might be uneven. So intermolecular forces have other complications or other examples as well. One example, when I was talking about water yesterday, I said, hopefully you all took a shower yesterday and you got out of the shower, the water was sticking to you. When that water sticks to you, it adheres to you. It's not water that it's sticking to your skin. So that's an example of intermolecular forces, one water molecule sticking to your body or multiple water molecules. Those water molecules also come in droplets. So those adjacent water molecules, intermolecular, they stick to one another. Those are examples of adhesion and cohesion. Intramolecular forces, adhesion and cohesion, what does that do to? Here's an example of what it's due to. First of all, we have 1, 2, 3, 4, 5 separate water molecules. Those 5 water molecules all have these slight negative and slight positive charges on them. And all you're doing here is organizing them as according to magnets, like magnets. So how do you do that, right? So if you had a magnet and I had a bunch of iron filings would organize themselves. Negative charge would be in one direction, positive charge would be in another, and that's what's happening here. You have positive charge of one water molecule and a negative charge of an adjacent water molecule, forming a temporary bond called a hydrogen bond. The hydrogen bond that I'm pointing out here occurs in water, and it results in intermolecular forces, intermolecular and intramolecular forces between adhesion and cohesion. It also happens in proteins. It happens in carbohydrates to a lesser extent, but certainly in nucleic acids. So hydrogen bonds are incredibly important. I mentioned that water is vaguely important for life. It's so important for life that astronomers literally will take probes and fly around the cosmos and look for planets with water on it. If there's water, then it's close enough to a star, then it's possible that there would be life, at least from what we could perceive. The reason for the importance of water, you okay? The reason for the importance of water is its ability to dissolve things. And when you dissolve things in water, that's called solubility. So when it's soluble, you take something and you separate it. And I'll give you an example up on the board right here. Sodium and chlorine, join them together, produce sodium and chloride. You place it in water and it disappears. So in contrast to other experiment where I took water and I took cooking oil and put it in there, that cooking oil separated to the top, the salt disappeared. It was still there, but it dissociated, separated. The sodium went to one area of the water, the chloride went to another area. The sodium went to the negatively charged portion of the water molecules, and the chloride went to the positively charged portion of the molecule.
So is that different from like fats separated from like water?
So oil and water are separated. That's exactly what a fat is. So oil, cooking oil is a fat. It's just a different type of fat, right? So body fat, right, is an example of a lipid, and that's just a solid version of cooking oil. It's a different chemical formula than cooking oil. But it's still that hydrocarbon, long chains of single bonded carbons adjacent to one another. Once in a while, there's a double bond in there.
Is it just hydrophobic?
And those are hydrophobic, right?
Just get another color.
So hydrophobic and hydrophilic, we talked about those. Hydrophobic are molecules that are non-polar. Hydrophilic are molecules or portions of molecules that are polar. If they're polar and they have transferred electrons all the way from one to another, you have an anion or a cation. When you say, if you drink Gatorade or some other of those Powerade and other things, you'll have them talk about electrolytes. Those electrolytes are designed, well, hopefully they're designed to replace them in your body. But honestly, if you're a normal, healthy young adult, you probably don't need, if you're exercising vigorously, if you're sick, if there's some other medical issue, right, you might need electrolytes, but otherwise you probably don't need it that much. So chemical reactions here, I'm not going to worry about chemical reactions in the context of this thing, especially this cell respiration and metabolism. We'll talk about that for next chapter.
I was gonna say, does that, I went to the doctor's once and they were like, that I should eat, potato chips so that the water in my body can stay, something like that? Is that like electrolytes?
So potato chips I wouldn't suggest that you eat potato chips. I'm not a physician, but potato chips aren't terribly healthy. But what potato chips would do is put additional salt in your body, and that salt will then prompt your kidneys to not release as much water because you have more salt in your blood. And so that's what's happening there.
Is that like, is that what
And salt is electrolytes, that's right. Sodium chloride, right, sodium chloride is an electrolyte, yes. So I have mentioned, we'll talk very, very quickly about 4 different types of chemical reactions. You've already seen 3 examples of one example. If you look at number 1, synthesis reactions, we will focus very heavily on that. Now, I'll be with you in 1 second. Synthesis reactions build things up. Decomposition reactions break things down. I'm going to tell you in a minute here, and I'm going to show you pictures on what I've already drawn that will take monomers and will make a dimer or a diatomic molecule, and then we'll make bigger and bigger molecules. And then if you do the opposite, you break it down. For those of you eating potato chips, that carbohydrate is going into your stomach, and then it's getting broken down slowly but surely. It's going into a carbohydrate, and then it'll get absorbed into your blood.
How is it that when you just said salt in potato chips is like sodium chloride, which is like electrolyte, but you get dehydrated, so how is electrolytes hydrating you if you're getting dehydrated from the sodium?
So it depends on the amount. Right? So your kidneys are going to be eliminating water on a regular basis, on a constant basis. Your kidneys are functioning normally. And basically, it gets to the urinary system the way that this thing called osmosis and active transport versus passive transport. But it gets to that type of stuff, and that's beyond what we're talking about today. Okay? So basically, that's a question for AMP2. Okay. So we're going to talk about 2 chemical reactions. The first 2, synthesis, you already know. Decomposition is also called hydrolysis. And I'll talk a little bit about that, but you should be able to recognize what's happening there. And then we'll talk about reversible and oxidation redox reactions in very, very little detail. I've already given you an example of a reversible reaction earlier today, and I'll show you what that is in a second. So if you look at the first example is dehydration synthesis or dehydration reactions. This is a synthesis reaction. And if you look at this picture right here, here's a question that I can ask. What is this? So it's an amino acid, but can you tell me which amino acid it is? Well, look at right there, that's R. Why is that 1? Well, there's 2 here. There's 1, 2. So this is 1, and that's 2. That's all they mean. Right? So if you look at this chemical formula and this chemical formula, they're the same. And what did they do here? Look at this, there's an OH and an H, we're taking that out. What are we putting? We're putting it over here. So it's just what I drew. So this is an example of 2 amino acids making a dipeptide, right? That chemical, that bond right there is called a peptide bond. And then if you do this 1,000 times, you would get a protein, or perhaps, or at least a polypeptide. Okay, so this is a dipeptide. Di means 2. Poly means many. And then you have strands of amino acids that can be adjacent to one another, that stick together, and then that could be functional protein, and we'll talk about that. So here is an example of synthesis reactions. Adding those 2 elements together, you're removing water, you're dehydrating it. If you look at this picture right here, you're doing the exact opposite here. On the left-hand side, we have what's called a disaccharide. If I show you this picture right here, oh no, you've never shown me this part. Yes, I have, right? This thing right here is called a glycosidic linkage. This is one monosaccharide, this is another monosaccharide. Together, because there's a glycosidic linkage, we have a disaccharide. Di is 2. So there's a disaccharide added in water. You add water, you put OH on one and H on the other. H on one, OH on the other. What do you get? You get a monosaccharide and a monosaccharide. By the way, there are multiple monosaccharides that you might have to recognize. I mentioned one earlier today called a pentose sugar, either ribose or deoxyribose. I'm not going to ask you to tell me the chemical formula, but this one I definitely will. If you look at this, 1, 2, 3, 4, 5, 6 carbons. There's 6 oxygens and there's 12 hydrogens. C6H12O6, the chemical formula of glucose.
So, I know this is like a lot of information that you're giving us. I don't know if you want to do this or you can, but at the end of the class, can you like make a list of things we should study?
So, here's a rule. If I talked about it yesterday and I'm talking about it today, should you study it? Am I going to waste your time talking about it? 2 or 3 times? No. Okay? So I can't do that for you, so that's your job.
Okay.
Arlen.
Do these chemical reactions apply to everything or just the volume?
Do they apply to everything or what?
Or just the volume?
Well, so when you're making macromolecules, you'll make them in the body. Generally, macromolecules won't be made outside of the body. So macromolecules are hard to maintain. They break down really easily. Right? So that if any of you are interested in farming or anything like that, if you take, like if you eat a salad or eat a sandwich or something like that, you take all that and you put it into a little bucket and you throw it out in your backyard, right? Are you ever gonna wanna go back and eat that? The same elements are there, right? They're reorganized a little bit differently. Right? Those macromolecules have then been decomposed, they've been broken down, and there'll be worms and other bugs in there, right? But then you can then take that compost and put it on plants, and the plants will grow very rapidly. They'll use that, and then you can eat it eventually. But it's the same elements there. Nothing's going to be lost necessarily. It's just reorganized a little bit. Okay, so the macromolecules are really important. So those are first 2 chemical reactions: dehydration synthesis and hydrolysis. They're opposites of one another. A reversible reaction, here is one example of this, is if you look at this thing right here, this is an example of a reversible reaction. So whatever on the right, you can go back to the left, and back from the left to the right, right to the left, and so on. I did this one earlier today. It's just a different way. This is just a different way of writing that. So we have oxygen and carbon dioxide. Oxygen and carbon dioxide. They can form carbonic acid right here, this acid right here with this little hydrogen floating around. If it's too high a concentration and it's in your blood, that could indicate a very serious illness. If it's not in your blood, right, if there's not enough carbon, most of the time there's not a lot in there, in your blood. So what does that mean? Your pH is about 7 in your blood. A reversible chemical reaction. If you're on the right side, you're going to go to the left. If you're on the left side, you're going to go to the right. okay? And it all has to do with the concentration on one side or the other. And then the last one is this oxidation and reduction reaction. This is a general term. What this generally means is that you're moving electrons from one compound to another, or one element to another. When the element receives the electrons, it gets oxidized, and when it gets loses the electron, it gets reduced. So reducing in charge or oxidizing. So oxidation and reduction here. I'm sorry. Oxidation is loss of an electron. I think I said that. Reduction is gain of electron. You're reducing the charge on the element so that that hydroxide compound, this chloride, has been reduced, it's gained an electron. This has been reduced, it's gained an electron. How do I know that? That negative charge there. The companion there is oxidation, those are going to be cations. We're not going to talk about energy, we don't have that. We don't have time to talk about that. Except to say, when you're worried about chemical energy, you're worried about ATP. That P is phosphor, or that phosphate group, and all you're doing there is you're taking that nucleic acid, which has two phosphorus groups, or phosphate groups, and you're adding a third. That third one is chemically reactive, and that can then do things in your body. Have we ever talked about homeostasis?
Yeah.
In the previous chapter, we have. Homeostasis is critical to maintaining life, to normal functioning life. Heat is generated by our body, especially if we're warm-blooded animals. So a lizard or a turtle do not have warm blood. They don't have the ability to maintain or for homeostasis of their body. They need external heat to come in and warm them up. We are able to do that by simply generating heat within our muscles. As a result of that generating heat, that friction there causes heat to increase inside our body. And then if we have too much heat inside our body, we lose it. If we don't have enough heat in our body, we try to maintain it. That's homeostasis, and that generating heat energy is through a process called diffusion, and we'll talk about diffusion in the next chapter. We're not going to worry about the speed of chemical reactions, but generally speaking, if there's a high concentration of these compounds, the rate will go up. If there's lower concentrations, the rate will go down. So we're going to skip this part right here. I haven't used this term inorganic chemistry, but the prefix in means not. In is not, and then organic chemistry. So inorganic chemistry involves the study of elements that I'm not terribly interested in. They're involved with the stuff, for instance, metal ions, potassium, sodium, iodine, bromine, fluorine, chlorine. So things that may have some biological importance, but not much. The things that I'm extremely important on are carbon-containing substances that have some or many carbons in that molecule. Those are called macromolecules. When you start forming macromolecules, you start from very small things called functional groups, and then you add to them and you build them up bigger and bigger, a little bit like a Lego, and then you build it bigger and bigger, and then you get a big thing. And that big thing is called a macromolecule. So that's the study of organic chemistry. And then more general, another term there would be biochemistry. Regardless of what we're talking about here, without this thing, we don't exist. I've talked about water in a couple of different examples yesterday and today, so we're not going to worry about that. So I'm going to scroll through this stuff right here, what some of the things that water does, including maintaining body temperature and things like that. Water can dissolve things, we've talked about that already, and if it's hydrophilic, then it will dissolve in water. If it's hydrophobic, it will not. I don't talk about solution or osmolarity at all. We talked about acids and bases earlier today. When you have an acid or a solution, the two elements here, or the two ions, are the hydrogen ion, or the proton, and the hydroxide ion. When the concentration of those two are about the same, the pH is at 7. When one is higher than the other, then it's not 7. The further away you get, either the higher or the lower the number is. So it's all basically in relation to the concentration of hydrogen or proton and hydroxide ions. And that's what the basis of acids and bases are. Closer you are to 7, the weaker the acid is, or the weaker the base is. The further away you are from 7, the stronger the acid is, or the stronger the base is. We're going to be particularly interested in acids. And if you look right here where my pointer is, this is what I just said, right? So if it's at 7, then it's the concentration of those two ions are the same. If it's very low and the pH is low, 1 or 0 or 2, then the concentration of hydrogen ions is much, much greater than hydroxide. And then if the pH is high, 13 or 14, the concentration of hydroxide is much greater than hydrogen ions. Here is the pH scale, a different way of thing. If you look at some of these examples right here where my pointer is so this graph right here is the pH on the left side and then the concentration of the two ions on the right. On the bottom. High concentration of hydrogen ions, high concentration of hydroxide ions, the same concentration of those two. Distilled water and blood is about the same. If we go way down here, lemon juice and stomach acid, hydrochloric acid, very, very strong acids. And then if you go up in the upper left, sodium hydroxide. Have we talked about sodium? Yes, we have. Sodium hydroxide is not a very good thing to be drinking, right, or to inhaling. Right? Why? What did we talk about? Elemental sodium here. Sodium is a highly reactive substance. Sodium hydroxide is highly reactive. The more, if you're at this end or in the upper left-hand corner, it's highly reactive and it's something that you want to stay away from, generally. Now, having said that, look at this. Stomach acid, highly reactive. I just said you want to stay away from it. Now, in this case, you want to stay away from it except in one place. Where is that? In your stomach. Your stomach is built so that it not only makes hydrochloric acid, it can contain hydrochloric acid, and when your body doesn't, is out of balance, is out of homeostasis, and you produce too much hydrochloric acid, then you can get something called an ulcer. Literally what happens is your hydrochloric acid eats through your stomach and eats a hole in your stomach and then perhaps the rest of your digestive system. That's a problem, right? So what has to happen? Well, you have to go through some changes there. So this is a highly reactive substance there, but it's designed to be in the stomach, and then once it gets out of the stomach, it gets, the pH goes way up in that stuff and it goes to almost neutral. So if you look at pH in blood, if we say that pH is normally around 7.3 to 7.4, 7.35 to 7.4, anything less than 7.35 is something called acidosis. Anything greater than 7.45, alkalosis. What is this an example of? We already talked about it. What'd you say? So in chapter 1 we've talked about it. So what I'm telling you now is that your body, when it gets close to 7.35 pH, what does it do? Does it allow it to go less than that? No, you want to get away from acidosis. So what happens? The body increases the pH. Then your body goes let's say 7.46, what's gonna happen? You're gonna let it go higher? No, because you're in alkalosis. Then you're gonna go down closer to, lower. What's that called? Which one? Not positive and negative.
No, negative.
Okay, it's one of those two.
I think it's positive.
If you want it to go up, you have to go down. If you want it to go down, you can.
Negative?
Isn't it the opposite?
So maintaining your body conditions in between these two set points, right? That's called negative feedback. Okay? If you're outside of that range, you're sick. Right? You're in serious health crisis. Right? And that's a major problem for many, many different organs, not the least of which your blood is going to be having a problem with oxygen maintaining, or amount of oxygen in your blood. So that's an example of negative feedback. If you're talking about positive feedback, if your pH rises to 7.45, positive feedback would then push it up to 7.5, 7.6, 7.8, 8, and so on. Right? That's not good. So you're not going to be that far away from this set point behind me and survive, right? Unless you have extreme medical procedures done, right? On a normal day. So here's an example of negative feedback. And we talked a little bit about salts and sodium, potassium, and sodium chloride. I don't talk a lot about different buffers. One example of a combination of two things, oxygen and carbon dioxide. When we talk about gases in our body, the two gases that are very, very important are oxygen and carbon dioxide. Sometimes people get confused. Where does oxygen come from? Well, let me ask you this. Where does oxygen come from in your body? And when I say oxygen, I mean O2, diatomic oxygen. Where does it come from?
From the atmosphere?
From the atmosphere. And where does it go to?
Your lungs?
Your lungs. And then from there, where?
Your blood?
Into your blood. And where in your blood?
To the cells?
To the
Which blood cells? We don't have red blood cells. What do we have?
erythrocytes,
We go to erythrocytes. And then from erythrocytes, where does it go?
hemoglobin,
So erythrocytes have hemoglobin. Where does it go? So blood is oxygen being carried by the blood via hemoglobin. Where does hemoglobin carry it to?
the tissues,
Your bones?
To your bones or your tissues, and then where?
The bloodstream?
What is the basic? No, you're already in the bloodstream. In your cells. Right? Now, your cells are going to undergo metabolism. They're going to break down macromolecules. They're going to break down carbohydrates, proteins, lipids, and they're going to produce a gas. And what gas are they going to produce?
Carbon dioxide?
Carbon dioxide. There's one element or one part of our cell that produces carbon dioxide. It's called the mitochondrion. The only place that carbon dioxide is produced is in our mitochondrion. There's other things a little bit, but generally speaking, it's carbon dioxide. That metabolic waste then does reverse. It goes from the cells, goes exported to the blood, gets carried from the blood to the lungs, and then it gets exhaled into the environment. So one of the things that I'm going to go throughout the semester is tell you is that I want you to always answer questions in greater and greater detail. Okay, if you sort of just generally answer the question, right, then we're going to go in further and further and further detail. This week in mini practical 1, you're going to see that as an example. Have I ever drawn any of these compounds?
Yes.
You should remember that whenever there's an intersection between two lines, you're assuming that that's carbon. If the intersection between two lines has things hanging out there, you're assuming those are hydrogens. So then this one right here that I'm pointing to is C6H12. This is CH4, that's easy. And then on the right, this is C6H6. The reason why that's C6H6 is the bonding patterns of carbon and hydrogen are 4 carbon bonds for each carbon atom. So this one right here has a pair right there. There's 2 chemical bonds. There's a 3rd chemical bond. And so that means that there's only room for 1 more chemical bond. I talked about functional groups yesterday, and I said I particularly focused on these 2 functional groups, hydroxyl and carboxyl, and particularly when I was drawing an amino acid. And I said there's, and then an amine group. And I said there's an amine group and there's an acid group. So the amino acid, the amine always has an NH2, and you see where that R is right here? That means there's something else hanging out there. This carboxyl group has a carbon with a double bonded O and an OH, or an alcohol group. And here's that alcohol, or OH. Whenever you see an R, an R, an R, or an R, that just means there's probably a carbon there and there's other stuff. So here's 4 examples of functional groups that I've talked about, whether I recognized it or not yesterday. There's hydroxyl, carboxyl, amino, or amine, and a phosphate group. Okay? Incredibly important in building macromolecules. There's others as well that are not nearly as important, but you might see them. For those of you who have long hair, I'm jealous. For those of you who have curly hair, I used to have curly hair, believe it or not. But where? No. If you have curly hair, then you have lots of that molecule right there, the S in there. Yes. So that's called a sulfhydryl group. So sulfur present in in keratin, in that molecule there, will form chemical bonds here, and it'll cause the hair to kink up, to curl up. So that's just the present. So sulfhydryl means that there's a functional group of sulfur. I don't think that I ask about carbonyl and ester groups. They're just different variations on that. And if you get into organic chemistry, and when you get into organic chemistry, you would get into that. Question?
Is that stuff what's inside of the pen?
You gotta yell.
Is that chemical there? That's what's in the pen to make your hair curly?
Oh, so the sulfhydryl?
Yeah.
Yes. Yeah. So that you're gonna basically make those new chemical bonds there. Yes. Have I ever talked about 4 macromolecules?
Yeah.
Okay. So the question should be, is this familiar? If it's familiar, and I hope that it is, then we're going to go through this quickly. Carbohydrates, monosaccharides. If you look at this picture right here, this is fructose. Now, I will not ask you to identify the name of fructose. I would only ask you to identify this one right here that I'm pointing at. That's glucose. Look at the chemical formula, C6H12O6. So in this class, I'm lying to you. Okay. In this class, we only have 1 macromolecule, 1 monosaccharide, and that's glucose. But there's lots of different 6 carbon sugars.
I don't understand that. Is it like all with the same like elements, just in different forms?
Yes, exactly. So on the left is fructose, on the center is glucose, on the right is galactose. Right? The only one that I would ask you to identify would be glucose. I could ask you to tell me the chemical formula on the left or the chemical formula on the right, and if you look at the bottom, they're all the same. So these are called isomers. I don't care if you know that. I do care if you know about glucose. If you take 2 monosaccharides on the left-hand side, we have 2 monosaccharides. It happens to be glucose and fructose. You make a disaccharide. When you take glucose and fructose, you make sucrose. You make that glycosidic linkage, that's dehydration synthesis. That oxygen right here is that glycosidic linkage. As a result of that chemical reaction, you're going to lose a water molecule. And then you can build that up very, very high. You can build that up really, really big. Carbohydrates come in 4 varieties. There's 2 in plants, 2 in animals. Two in plants include starch and cellulose. How many of you like to eat corn or potatoes or potato chips, right? How many of you would eat paper or wood or other stuff like that, right? So you will be eating the same thing. The only thing that's different is that you're rearranging those monosaccharides in a different organization. One is tasty, the other is not. If you end up eating lots of fiber in your diet, like salads and lettuce and stuff like that, what you're eating is a lot of cellulose. So there's starch and cellulose that's formed in plants. Two types of carbohydrates formed in animals include glycogen and chitin. Chitin is a structural compound, very, very hard compound found in crustaceans, lobsters, and crabs, insects, and mushrooms. So that is something that you can't digest either. Cellulose is something you can't digest, so if you ingest it, if you eat it, it passes out of your gut unchanged. Glycogen is a carbohydrate, is a storage molecule that you find, you store in our bodies, or as a form of energy. So basically, all you're doing here, if you look at this picture right here, here's one monosaccharide, here's another monosaccharide, a third monosaccharide. Each of these monosaccharides are joined by glycosidic linkages. Whether it's going vertical or horizontal, they're all glycosidic linkages. And how do you do that? Through dehydration synthesis. You just have to do it again and again and again. Triglycerides come in a number of different categories, but lipids. The one that I drew is a triglyceride. On the left-hand side is glycerol, right here. And we have 3 fatty acids. And in this case, these 3 fatty acids are undergoing dehydration synthesis, and we're going to make a triglyceride. Now, if you look at these 3 fatty acids, they're all identical. They have the same number of carbons, the same number of hydrogens. That's not always the case. So if you look at this fatty acid, this is called a saturated fatty acid. And if you look at this fatty acid, it's sort of wiggly. Why is it wiggly? Well, there's double bonds here. There's a double bond there, and a double bond there, a third double bond. There might be many, many, many double bonds. The more double bonds there are, the wigglier it gets. The wigglier it gets, it just changes the chemical nature of it. Right? So that's called an unsaturated fatty acid.
Is DNA just full of double bonds? That's what it's
That's at a different level, right? So the reason why DNA is in that is the structure of the nucleotide, and we're going to get to that. Okay? So lipids come in triglycerides and phospholipids. Now, if you look at this picture on the left-hand side, we have a phosphorus group at the top. There's a glycerol molecule that's not labeled. And then we have one fatty acid that's straight, and then another fatty acid that's wiggly. That fatty acid is wiggly because it's unsaturated. It's got double bonds in it. If you look over on the right-hand side of this picture, this part right here is a fatty acid. Again, it's bent, it's unsaturated. The right-hand side of the fatty acid is straight. This is saturated. Okay, so now there's 2 fatty acids, and then up here is the phosphate group. So this is a phospholipid, and I drew a phospholipid almost like this, but I had a little bit, what I did was in my drawing of the phospholipid, I erased one of these, and I put the P on the other end of the glycerol. So in that case, then, the P on the other end of the glycerol, because it changes the chemistry of this triglyceride will be hydrophobic. These 2 fatty acids will be hydrophobic because there's lots of equally shared chemical bonds, and they're nonpolar. Anytime you see carbon and hydrogen, it's nonpolar. Lipids are nonpolar, except for these. This part right here has one area of the lipid is called phospholipid. That phosphate group is polar and it's hydrophilic. The tails or the bottom are non-polar and they're hydrophobic. So the bottom of this picture acts like a lipid, the top of this picture acts like something else. So if you, I mentioned earlier or yesterday, there's different types of lipids in our body. Here is 3 examples or 4 examples. So on the upper left-hand corner, we have cholesterol. In the lower, on the right-hand side, you have estrogen and testosterone. I don't think that I asked questions about bile, which happens to be in your liver, produced in your liver and stored in your gallbladder. But I do ask questions about estrogen and testosterone. Look around the room. Some of us may have more estrogen versus testosterone. Others have more testosterone versus estrogen, right? I don't care about that. What do I care about? If you look over here on the left-hand side, this is a lipid that is definitely differently shaped, right? It's not something I've drawn previously. But if you take this and you modify it a little bit, you make estrogen. And if you take this and you modify it a little bit, you make testosterone. Okay, so this is the basis upon which your building block, and it is definitely a lipid. It's called a steroid. Amino acids. Have we talked about this? We have. Right. Peptide bond, dimer, right here. Here's a bunch of different examples of amino acids. There's 1, 2, 3, 4 different amino acids. The simplest amino acid is the one that I might ask you about, and that's where my pointer is, called glycine. Glycine has just 1 hydrogen hanging off of there in that white box, which is indicated right here. So I will show you this amino acid and ask you to identify it. I could show you this amino acid, which is the generic amino acid. If I were to show you any of these other amino acids, I wouldn't ask you to identify it. I might ask you to tell me what type of macromolecule is it, or what is a monomer of it. So these are amino acids. How do you know it's an amino acid? What's on the left? NH2. If you see NH2 on the left, and then COOH on the right, that's the amino acid. The right side is the acid, the left side is the amine group. Okay? No matter what you have in this white box or this white box, it doesn't matter. There's the amine group and the acid group. So you have to pay attention to what you're looking at. Here's it in greater detail, and just to illustrate for you, I could put this picture right here. It happens to be a tyrosine. Cross out this. What monomer would this be of what monomer of the macromolecule would be? Protein, carbohydrate, nucleic acid, or lipid? It's a protein. What type of molecule is it? It's an amino acid. What's this functional group? It's an amine group. What's this functional group? It would be either an alcohol or a carboxylic acid group. I wouldn't ask you anything about this as far as the chemical formula, because that's the R group. Okay? So once again, if I spent time drawing it, then I might ask it.